Let's get started on Chapter 1, which will be an introduction to microbiology. The whole course will be dedicated to studying organisms that we can't see with the naked eye. The types of microorganisms that we'll be discussing are bacteria, archaea, protozoa, fungi, helminths, which are worms, viruses, and algae. As far as studying these organisms goes, it's easy and difficult for various reasons. They can reproduce within a matter of minutes to hours. Many can easily be grown in the lab. However, a lot of them require the use of microscopes and/or special staining procedures. These microorganisms are not new inhabitants of Earth. They've been around for billions of years. You may remember your 3 cell types from 1406. These guys all came from an extinct common ancestor. They include eukaryotes, which are organisms that contain a nucleus, bacteria and archaea, which don't contain a nucleus. And this particular figure is a phylogenetic tree, which is based on genetics, shows relatedness between organisms. You can see all 3 of these organisms originated from one last common ancestor. When we look at bacteria and archaea, these guys are pretty simple. Single-celled organisms, whereas the eukaryotes are a bit more complex, but they can also be single-celled as well. They're typically larger, but are smaller in numbers compared to those prokaryotes in bacteria and archaea. In this evolutionary timeline, you can see that the prokaryotes and eukaryotes, or the earliest of the eukaryotes, have lived together for a good while, and that we as humans, if we consider this to be a clock, we take up about seconds. So kind of mind-boggling there. What does this term ubiquitous mean? It's a fancy way of saying everywhere. We can find microorganisms virtually anywhere in the world, as you can see here. Most microorganisms are given a bad reputation, but we will talk about how some are essential to life. You may have heard the term evolution before, so let's discuss that. Evolution describes changes seen in organisms so that they can adapt and survive in their environments. It is a theory, which means there's a substantial amount of data that supports it, and it is testable. It's not the only theory in science. There are plenty of others out there. Of course, we know that plants can undergo photosynthesis to convert carbon dioxide to sugars. It may surprise you to learn that bacteria were the first organisms to undergo photosynthesis. However, they did it without producing oxygen. Throughout time, organisms evolved to perform oxygenetic photosynthesis, which is what we're familiar with. These microorganisms actually perform photosynthesis at a larger percentage compared to plants. These microorganisms that we will be discussing this semester do everything, or contribute to everything from soil and water to the atmosphere. They're responsible for producing gases that make up our atmosphere, are heavily present in water, and also live symbiotically with plants. We as humans have been using microbes for years, including but not limited to using yeast to make bread and alcohol, making cheese, using breads, believe it or not, to treat wounds, using these microorganisms for mining purposes, And bacteria can also be used to clean up oil spills through bioremediation. There have also been a lot of technological uses with microbes as well. Through genetic engineering, the genetic makeup of organisms can be altered. These genetically modified organisms can be used for good, such as production of pharmaceutical products. This can be used alongside recombinant DNA technology. As I mentioned before, bioremediation uses microorganisms to clean up toxic spills. Microorganisms typically are given a bad name, but the majority are harmless. Those that do cause disease are called pathogens, and there are roughly 2,000 different types of microbes that fall under this category. These infectious diseases are the most common causes of death worldwide. If you look at this picture of the top causes of death, we have diseases caused by microorganisms depicted in fuchsia, and those diseases that are not currently known to be caused by a microbe, depicted in blue. Here in the United States, heart disease and cancer are the top causes, whereas worldwide we can see lower respiratory infections making the top 4. This can likely be attributed to the lack of access to medications. Throughout this semester, we will discuss emerging diseases, which are either new diseases or diseases that used to be found in isolated regions and now spreading. Re-emerging diseases are those that were under control, but perhaps travel as well as anti-vaccination has contributed to their return. There is now research that is linking non-infectious diseases to certain microorganisms, such as stomach ulcers linked to a bacteria. Throughout the world, we are seeing increased numbers of patients with immune systems that aren't functioning properly, whether it be something they're born with or acquired. This makes them more susceptible to disease. We are also seeing microorganisms that are becoming resistant to typical methods of treatments. Let's look at the organization of our 3 major cell types. Eukaryotic organisms contain organelles, which are basically miniature organs with their own functions. Bacteria and archaea are significantly smaller, don't have organelles or a nucleus, and require a microscope to observe. Viruses, the story of the past few months. Viruses are a completely different ballgame, and there is a whole chapter dedicated to them. These guys are not true cells. They are extremely small, but still contain genetic information, whether it be DNA or RNA. Outside of a host, they lack the ability to reproduce, but they do contain a few genes. We will get to this in detail later. The 6 different types of organisms that we will discuss are depicted here and shows the relative size differences. Let's talk about this idea of spontaneous generation. Long time ago, people believed that life arose from thin air, sort of like magic. People set out to disprove this silly notion, but even after the data was there, abiogenesis still prevails. Biogenesis is the complete opposite of spontaneous generation and states that life has to come from life. Louis Pasteur was important in disproving spontaneous generation. On the next slide, we will discuss his swan neck experiments. Pasteur started off with 2 flasks, and he used heat to sterilize the broths. It killed any organisms that may have been present. He then broke the neck of one of the flasks, which exposed it to the air. He noticed that in the flask there was a cloudiness to the broth, indicating the presence of microbes. In the other flask, he left the neck intact, and in that broth there was no growth. This proved that even though we can't see the microbes in the air, they're still there. It proved biogenesis, that life came from life. Robert Hooke was vital in the identification of cells. Antony van Leeuwenhoek is the father of microscopes and was the first to identify bacteria, which he described as animalcules, which he saw from the scrapings of friends and family members' teeth. Kohn helped to identify endospores, which we will discuss further in a later chapter. Holmes and Semmelweis helped to enforce hand washing to prevent disease, which we've been hearing about plenty in the past few months. Lister developed procedures to minimize infections in surgical units. Of course, we discussed Pasteur and disproving spontaneous generation, but he also developed pasteurization and helped to link disease to microbes. Robert Koch was vital in determining that microorganisms can be linked to diseases. He specifically linked the bacteria Bacillus anthracis to anthrax. Restriction enzymes were identified in bacteria and can be used to cut DNA at specific locations. They have been used in genetic engineering and medical interventions. The polymerase chain reaction came about in the 1980s and is a means to amplify DNA and make billions of copies. This is something we'll look at a little bit more in detail later as well. Small RNAs are relatively new. There's only a small percentage of DNA that codes for a protein, and those regions that don't are small RNA. Their importance in regulating cell activity and can be used in cutting-edge treatment for certain diseases. The Human Microbiome Project helped to identify what types of microorganisms live in and on us. This has been great to see differences among individuals and to identify alterations that may be associated with diseases, such as cancer, as well as depression. Let's discuss our 4 major macromolecules. These carbohydrates, lipids, proteins, and nucleic acids are all polymers made up of monomers, each one, of course, having a specific function. This table outlines all the major macromolecules and examples of each. This is a good study tool where all of them are outlined in one location. Don't worry about memorizing everything, just hit the major points. Carbohydrates are your sugars. Some important ones being your pentose sugars, ribose and deoxyribose, along with glucose, which is utilized to synthesize ATP. There are various types of polysaccharides found in all types of microbes. Cellulose can be found in plants and algae. Peptidoglycan is a major component of the cell wall of a bacteria. The lipopolysaccharide is found in gram-negative organisms and contributes to their pathogenicity. Some bacteria secrete a glycocalyx, which is a sticky substance that enables them to adhere to surfaces. Lipids are your fats. Triglycerides are important in storage as well as energy sources. Phospholipids are of major importance in the cell membranes of organisms. These lipids have a water-loving head and water-fearing tails. The difference in charge and behavior allows them to form a protective bilayer. Here we can see the structure of the lipids. Those water-loving heads on the outsides and the hydrophobic tails in the middle. Steroids are also important components of the cell membranes of some organisms and also exist as hormones. Cholesterol plays an important role in bacteria who lack a cell wall. Waxes, as far as microbes go, are found in bacteria associated with tuberculosis along with leprosy. It is something that makes them more difficult to treat. Here you can see the cholesterol, which is embedded, get my mouse, which is embedded in the cell membrane and helps in reinforcement. Proteins are the guys that do it all. Their building blocks being amino acids. A lot of times you'll see polypeptide and protein interchangeably used. There are 4 different structures of proteins, primary being like beads on a string, while secondary involves an alpha helix along with a beta-pleated sheet, which are formed by hydrogen bonds. Here you can see both your primary and your secondary structures. In the tertiary structure, we see the formation of more bonds between the functional groups of the amino acids. Your quaternary structure involves multiple polypeptides combining. You can see the differences between the tertiary structure and quaternary structure. Here we can see at least 3 polypeptide chains. Every protein is constructed differently, and they each have their own shapes and can only bind at specific locations, almost like a lock and key. A couple of important proteins are enzymes, which speed up chemical reactions, and antibodies, which are part of our immune system. They are responsible for binding to specific locations on microbes. The native state of a protein is what we typically see, and one which performs normal functions. There are several things that can alter that native state, including heat, alcohol, and disinfectants. By altering that native state, it can render the protein useless or slow activity. Our next macromolecule is our nucleic acids. DNA, which contains the set of instructions to produce proteins, and RNA, which assists in this process. The structure of DNA is a double helix, with the nucleotides being held together by hydrogen bonds. The nitrogenous bases have a set way of pairing. Adenine bonds with thymine, while guanine pairs with cytosine. We can see the double helix along with our nitrogenous bases held together via hydrogen bonds. You can see the pentose sugar along with the phosphate groups. RNA is single-stranded, and there are several types. mRNA is the middleman, involved in translation of DNA to proteins. tRNA brings in anticodons and amino acids during translation. rRNA makes up ribosomes, and you also have RNA involved in cell regulation. ATP is the currency of our cells. It is our energy source. The energy is stored between bonds of the second and third phosphates. It is when the bond is broken that the energy is released. So what do these different cells look like? We've discussed the prokaryotes being single-celled. Animals contain trillions of cells. These cells have different shapes, all contain protoplasm, DNA, and ribosomes. This slide sums up a previous slide in the chapter. How do we name organisms? Carl von Linné is the father of taxonomy. He developed a system to identify organisms. He identified a classification system to help in this identification process. It's a binomial nomenclature system containing two names. Both are italicized if typed and underlined if written. The genus is capitalized and the species epithet lowercase. Let's discuss the classification. Our most broad group is the domain, with the most specific being the species. We discussed the phylogenetic tree earlier, which looks at relatedness. Of course, throughout time, changes have arose in organisms. Initially, Darwin and Haeckel identified two kingdoms, plants and animals, with Protista and Monera being added later. Whittaker took it a step further and added fungi to the list. Woese and Fox developed the three major domains that we are familiar with based on genetic differences, these three being bacteria, archaea, and eukarya.